So hello everyone, this is a lecture, chapter 6 on enzyme kinetics, and I'm recording this lecture because earlier today the live lecture in the lecture hall at the VU didn't work out because the desktop computer didn't work. So that's why I'm recording it now once more, and it's going to be, it's the same lecture that I gave live this afternoon. So it's going to be about chapter 6. Enzyme kinetics. The first answer that we need to, the first problem that we need to look at is why do cells exploit enzymes? Well, the first reason is that many of the reactions that occur inside cells, if they would occur in the cytosol, so rather than in the catalytic side of an enzyme, they would hardly occur. And so in other words, the catalytic side of an enzyme offers a favorable physicochemical conditions for reactions to occur, such that they occur much faster in the catalytic side of an enzyme than they would in the cytosol. So enzymes offer favorable physicochemical conditions in their active site such that the reaction that they can catalyze occurs much faster in the catalytic site than in the cytosol. And the conditions in the catalytic site are determined by the local environment shaped by the occurring amino acids. Okay, so the precise amino acid biochemistry in the catalytic site now determines the physicochemical conditions at which the reaction is going to take place. And these conditions are different than the watery environment of the cytosol, so which is why enzymes can, inside the catalytic site of enzymes, particular reactions are more favorable than they would be in the cytosol. This is sometimes associated with the statement that enzymes lower the activation energy of a reaction so they speed up reaction rates. Now, the second reason why enzymes are favorable for cells is that by changing gene expression, so leading to changes in enzyme concentrations, cells can control which reactions occur, and how fast. So since enzyme reactions are going much faster than chemical reactions, so uncatalyzed reactions, so the only meaningful reactions for the cell that are occurring are enzyme catalyzed, because all the other spontaneous reactions that are occurring without a catalyst occur much slower. And therefore, by changing gene expression and hereby changing the concentration of enzyme, cells can control both the identity of the reactions and the rate at which they occur. Now, the final reason why enzymes occur is that enzymes allow for decoupling of energy demanding reactions to those that supply energy. So for instance, if you think about the import of glucose into a cell, and the intracellular concentration is larger than the outside concentration, then clearly glucose has the tendency to leak out. So the cell would no longer be capable of importing glucose, because that would occur against this gradient. Now, in order to make that possible, the cell would exploit ATP hydrolysis, and the energy that's available in that reaction, it can couple to the import of glucose to the inside environment. And now, two reactions, so glucose out to glucose in, which is reversible and would go in the reverse direction. So you would accumulate glucose outside if the intracellular concentration is greater than the outside concentration. But now, by coupling it to the ATP hydrolysis, this energy that is liberated can be used for import. And then you can effectively accumulate glucose even though the inside concentration is greater than the outside concentration. So coupling allows energy transfer between one reaction that's energy demanding. So this would be the energy demanding reaction, and this would be the energy liberating reaction. Okay, so that's the third reason. So now the next question that we need to answer, so why do we need enzyme kinetics? Since we can already describe enzyme mechanisms in terms of mass action kinetics. So let me give you an example. So if we are considering the reaction where two substrates are converted into two products by some enzyme E, so for instance, glucose plus ATP is converted into glucose 6-phosphate plus ADP, catalyzed by hexokinase. Possible mechanism, enzyme mechanism, for this enzyme catalyzed process would be the ordered binding of substrates. So S1 binds first and then S2 binds. Now you would have the catalysis reaction where the substrates are formed into the products in the catalytic side of the enzyme. And then you could have the ordered release of the products, liberating the same enzyme again. So since we end up with the same enzyme as we started with, the enzyme as a catalyst, and these are now substrate binding events. So this is catalysis, and this is product release. And this would be an ordered mechanism. And the other terminology is that each enzyme mechanism, in this particular case, it's composed out of 5 reactions. And these reactions are called elementary. So 5 elementary reactions. So the terminology is elementary, occur in this reaction mechanism. But this is only one possible mechanism, because there is an alternative mechanism. An alternative enzyme mechanism. It does not have the ordered binding, but it has a random binding. So for instance, this is possible, but rather than substrate 1 first binding, it could also be substrate 2. And then from that state, you can get the state where both substrates are bound. The catalytic event takes place. Now your P2, P1 can release first, or P2 can release first. So here you have ordered substrate, random substrate binding and release. And these two mechanisms, of course, are different. So here you have, for instance, 1, 2, 3, 4, 5, 6, 7, 8 elementary reactions. And we have 1, 2, 3, 4, 5, 6, 7 enzyme species or states. Whereas here you only have 1, 2, 3, 4, 5 enzyme states. So clearly these two mechanisms are not identical, and the task for an enzyme specialist, so an enzymologist, is to identify, is the reaction following the ordered mechanism, or is it following the random mechanism? Now, the problem in these type of identifications is, so the enzymologist is faced with, well, at least two problems. One, measurement of enzyme states, so the concentrations, is generally not possible. So we really lack the experimental methods to distinguish the 5 different enzyme states in this mechanism and the 7 enzyme states in the other mechanism. And the other thing is that also determining the rate constants of all the elementary reactions is problematic, if not impossible.